Uh, and thank you to the GSAC for organizing this seminar and give us some space to talk about our research. So today I'm gonna talk about the spiders, colors, and evolution. I hope I can keep you awake as I'm keeping myself here in Australia. That is already 2 a.m. Well, this topic that is really passionate and interesting. So ecologists and evolutionary biologists had focused largely on heritable visible marker phenotypes to explore phenotypic variation. For example, the beaks of the Galapagos finches that we are mostly familiar with, or the growing forms of this beautiful Australian plant with huge phenotypic variation on the way they grow. In one hand, because they are easy to measure or to follow, or because they are traits that change fast in time and in a small geographical scale. Body coloration is one of the most studied phenotypic markers because who doesn't like color? But actually because it is a trait that confirms survival and reproductive success through various ways, acting as a sexual trait, as you can see here with these beautiful Heliconis butterflies, or giving advantage for camouflage, protecting for predators, or protecting for predators as a aposematic trait, or offering physiological advantage via UV protection, antimicrobial protection, and thermoregulation. Coloration as a trait has been studied in species that present multiple color phenotypes within a single population, something known as color polymorphism. It is a common phenomenon in nature, and it has improved on our understanding on how natural selection operates and drives the evolution of traits. Despite color polymorphism being a common phenomenon in nature, research has been biased towards vertebrates and a few insects, which mainly use coloration as a sexual signal or warning to predators. And relatively few studies have focused on polymorphic lineages that rely on colors for other ecological purposes. For example, web-built spiders, they have really bad vision, so they don't use coloration as a sexual signal. Instead, they use their abdominal colors for camouflage, possibly as aposematism, because they are ectotherms. These spiders rely on coloration to reach extreme environments like really cold ones. Coloration in these spiders play an important role in foraging because they are zero-away predators, so they depend on these colors to deceive their prey, matching things that they like for example, flowers or other sexual individuals that they are interested in. So, as in this case. And this function can be related with the presence of multiple color variants in a single population. So I was planning to start my PhD, the COVID pandemic hit. So I was figuring out what to do with my time at home while everything was happening. So I decided to start answer this question as an start of my PhD. So actually spiders with bright colors capture more prey. So I studied population of the species Gasteracantha cantiformis, which is this beauty that you can see on your right. And it varies on their dorsal and ventral colors on a single side. So the side was this empty bike path near my parents' house. So we went there to examine the webs of these spiders from a lateral view to count the number of prey that they capture, while considering other variables that could affect prey capture, such as like luminosity, web area, or the size of the spider. So we did not find significant difference in prey capture rate between morph, which means that prey capture doesn't have a role in the presence and maintenance of within population color variation. Here you can see that the different color variants don't differ in that, and actually empty webs capture more prey than the ones with any spider. Instead, our data suggests that color morph of this spider may differ in their microhabitat preferences. So individuals with dark colors on their ventral side, here on your right, tend to be more prevalent in places with low luminosity compared with individuals with bright colors. So was something nice that we figured out from our data, and it's something that you can actually perceive when you are in the field. So it's possible that the presence of contrasting microhabitats in heterogeneous environments has promote phenotypic variation due to positive frequency dependent selection within each habitat. So it is possible that dark individuals here can find camouflage in shade areas, while bright individuals exhibit an aposematic signal where it can be more exposed to predators. An example of this multi-niche hypothesis is Timema stick insect walking sticks. So in this species, color morph occupy distinct ecological niches provided by the host plant, reducing predation and promoting color variation. So here you can see that the green individuals are on the leaves, while the melanistic ones are on the branches. However, a formal test for this multi-niche hypothesis in this spider remains to be done, but our data tell us that maybe this is what is going on. So this is like the general conclusion for this part. So now let's imagine that this scenario that I showed you here is located in the northern part of South America, like this. So because similar environments are also present across the continent, it is possible that a color polymorphic species like this spider could disperse because of their color variation. The probability of establishment in new areas can be higher, increasing its geographical range. And in fact, this spider is widely distributed in the continent with color polymorphic populations all over its distribution. However, we don't know if the wide distribution is a consequence of the possibility of using multiple habitats. But this association between color polymorphism and geographical ranges has been supported in groups like damselflies, butterflies, and vertebrates that, as I told you earlier, mostly use coloration as a sexual trait or as a warning signal. So we don't know what is going on with lineages that use coloration for other ecological purposes. So this question is if actually color polymorphism is associated with the geographical range of spiders came to us. So we expect that species with color polymorphism, like the one that I just told you about, have wider distributions compared to monomorphic species that we expected to be more restricted to certain areas. So to explore this question, I built a list of color polymorphic spider species, starting with a systematic literature research in Web of Science using the search term that you can see here. Then I searched for descriptions of color variation in the literature, especially the ones published by Helbert Levy, which had a work on a spider as vast as his eyebrows in this photo. Finally, I validated if the species that I found were truly polymorphic and discarded those that present geographical variation instead of within population variation, checking out the reports of these spiders on iNaturalist. So from this search, I found that there are at least 140 color polymorphic species of our web spiders, and they are mainly distributed into families, the family Pteridae here on your left, and Araneidae here on your right. And here are some examples of these beautiful color variable species. When we look at the distribution of where these species are, we discover that they are mainly distributed in temperate areas rather than in tropical regions, which was quite like unexpected. And as you can see here, the highest regions of color polymorphic species is in Europe. And this could reflect that the majority of the color polymorphic descriptions and also more geographical records are available in this area, meaning that we need to do more work in tropical regions. So I reconstructed the most recent phylogenies of the two families that I just told you about, or there are like big and diverse families, using genetic data available on the NCBI, and I included 41 polymorphic lineages that I found and confirmed that the species on the phylogeny were monomorphic or polymorphic using the literature available on the World Spider Catalog, that is this database and website with information about systematics, taxonomy, and a lot of like literature on spiders. And this is what I got. So here you can see that the color polymorphic species are highlighted in orange. And it seems that the presence of color polymorphism is widespread throughout the phylogeny. And there are some interesting groups that are quite rich on color polymorphism. Here is an example of these spiny spiders that I really like. There are multiple species that present this phenomenon. So I use this phylogeny to control for phylogenetic relatedness, and to answer my research question, I use multiple measures of geographic size, such as latitudinal range, area of distribution, the number of ecological regions occupied, or the number of climatic zones that they occupy. And here you can see that positive estimates on the effect of the presence of color polymorphisms are shaded in purple, while negative effects are shaded in red. Contrary to what I was expecting, no matter the source of evidence, I didn't find any difference between polymorphism and monomorphism. So there's actually no effect of the presence of color polymorphism on these measurements. As you can see, they mostly all of them overlaps, the estimates overlaps with 0. And these results contrast with previous findings that support the association of color polymorphism with broader range sizes. Nevertheless, we did find that color polymorphic species have a higher number of geographical records on islands compared to monomorphic ones. Here is the estimate, and you can see here the difference in proportions. So this might mean that for monomorphic lineages, it's more difficult to survive when they arrive on an island due to their reduced phenotypic variation. So if they jump to this island, given that they have no like, phenotypic variation in coloration, they might get extinct, locally extinct, while for color variable species could be more plausible to successfully conquer new areas, since a higher phenotypic diversity increases the probability of establishment in novel areas. Another way to interpret this result is that ecological opportunity and ecological release on islands can promote extensive microhabitat use that facilitates the maintenance of different color morph in the population. There are some examples that support this hypothesis in snails. And also, it has been suggested that ecological opportunities offering islands is a requirement for the huge phenotypic diversity of some adaptive radiations. So these are two explanations of the pattern that we found in this data. However, I want to mention and make emphasis that the results that I just showed you have to be interpreted with caution, because despite the fact that spiders are really abundant and diverse, it is the second group most neglected when considering geographical records. Here, you can see that it's just followed by insects, especially in the tropical regions. And this could explain the geographical pattern of abundance that I showed you in the map at the beginning, and the lack of differences in the geographical measures that we measure. So we need more people testing evolutionary hypotheses with the spiders. So maybe you get some inspiration from this. So the conclusions for this part are that color polymorphism is not associated with the geographical range of spider species. And this contrasts with previous findings in insects and vertebrates, and that color polymorphic species have a higher number of geographical records on islands, which could be an indicator of colonization success. From this work, you can also notice that there are multiple species of our web spiders that are color variable, and this offers an amazing opportunity to test multiple evolutionary hypotheses. Most of them seems to be reported on temperate regions, which means that maybe we need to study more these beautiful lineages in the tropics. So even considering that color polymorphic spiders are not associated with widespread distributions, there are some color polymorphic spiders that are widespread. One of these is the Christmas spider. So here in Australia, a bunch of species have the word Christmas on their name because it's when all the books are out because it's summer here. So yeah, this is an example of that. So taking advantage of this phenotypic variation and the widespread distribution means that these type of organisms are exposed to different environments. We decided to explore this question that is like, how is the climatic variation generating and maintaining the color variation in arachnids? So these spiders that I just showed you here is widespread throughout Australia, and it was reported in 1991 as color variable based on observations in Western Australia, and this is how they look in reality. But besides, it is really common. We have no information on how this color polymorphism changes across the distribution of the spider. So our first approximation to this is figure out the ecological and the effect of ecology and climate on this. So because of its coloration, we expected, so here you can see that you can find completely black or black and white. So because of this pattern, we thought that maybe the dark colorations confer some thermal benefits, which means that the dark ones should have higher heating rates compared to the lighter ones. And this would be reflecting in high abundance of dark individuals in environments with low temperatures. And when the temperature increases, this abundance should be reduced, and probably the abundance of the lighter individuals should increase. So first, considering that the dark colors can confer a thermal benefit, we explored using a control environment. So we use a solar simulator to replicate the actual power of the sun. And we put black and white black spiders inside this chamber that isolates the spider and controls for effect of wind temperature and wind speed. So within this chamber, all the climatic conditions were constant, and the only variable that changed was the color of the spider. So after heating up and frying some spiders in this setup, we found what was expected, that darker spiders have higher heating rates. However, we have no idea if these differences in heating rates are actually significant in the field. I mean, we don't know if after including variables like wind, speed, wind temperature, distance from the soil, actually this difference in body temperature and in heating rates will remain constant and actually mean something for the ecology of the species. So we decided to validate these results that we got in the lab, so we went to the field on a cloudless day at noon. There's a population here near Melbourne with hundreds and hundreds of spiders in only like 50 meters. So we start taking thermal photos like this one I showed you here of the spiders, so that let us calculate the body temperature. And we also measure the ambient temperature at the time we took the photos, the wind speed at the time we took the photos, the height of the spider, and we also calculate the size to control for all these things that can actually happen in the field that we didn't include in our lab experiment. So our field measurements reveal no temperature differences between the black and white morph on their dorsal or their ventral side. So here you can see that the mean estimations of body temperature of these spiders kind of like overlaps a little bit. So instead, we found that wind temperature and speed appear to be the common factor explaining the variation in body temperature in our data. So if it is not temperature, what is causing this color variation? Well, so we decided to explore more from a macro perspective. So we use like macroecology and a macroecological approach using different sources to collect geographical data. So I visit the main museums in Australia, checking out where the colors were reported. And I also spent a lot of time on iNaturalist, seeing what people were reporting. And because these morphs are kind of like discrete, they are really easy, to discriminate and to categorize. I also talked with a bunch of people on Facebook that were interested in spiders, and this was really, really helpful. And then with all this information, I validate all the data doing fieldwork. So I found that the black and white morph was the most abundant morph throughout the distribution. So you can see that it's everywhere, followed by the dark morph. And based only on geographical records, this morph coexists and be part of the distribution, but the dark morph seems to be absent in the north part of Australia that is more tropical. For my surprise, and this was really excited, I found a third morph that was considered like rare. The only reports were on this island. When you start going to inland Australia, this color variation start to be really common, and I'm gonna tell you more about it in a bit. So I'm gonna show you the difference in the climatic niche of these spiders using these density distributions. So it seems that in mostly all the variables that I consider, the black and orange morph are really different. So here you can see that the black morph tend to be more abundant in places with lower solar radiation, while the orange morph is in places with higher solar radiation. The orange morph is more abundant in drier places. But for example, they seem to don't differentiate in places with different minimum temperatures. So they seem to vary a lot of what they like. Also, an interesting pattern that emerged from this plot is that the density distribution of the black and white morph, that is represented here with yellow, is usually wider and covers the density of both black and orange morph. And in some cases, the density distribution is bimodal, sharing one of the modes with the other morph. So this was interesting to check out. So to measure the niche difference, we calculated something known as the D statistics, where 0 indicates no niche overlap between color morph and 1 represents complete overlap. So we found that the climatic niche of the orange morph was the most dissimilar, presenting low to medium overlap values when compared with the black and the black morph. Here, value around 0.2. And in contrast, the climatic niche of the black and black-white morph were more similar with high to medium overlap and value of 0.64, that is closer to 1. So it seems that the orange morph is really different. So this is all from a macroecological perspective, but we don't know what actually was happening in the field. So I'm gonna tell you about it, but then I want you to appreciate this spider for 10 seconds while I drink a little bit of water. And you can process all the information that I just shared to you. Okay, great. So, as I told you, this macroecological perspective doesn't tell us how actually the polymorphism is being shaped by the climate, because we don't know how the abundance is changing. So we decided to go to the field to measure abundance and more frequencies. So to explore this, I have the opportunity to travel a long way. This was a really long drive that we did during one season with the help of a lot of people to drive all these distance. So in total, we travel around 5,000 kilometers throughout the distribution of this species in the southern part of the country. So what I was looking for were big aggregations of these spiders. So I didn't tell you something about the biology of this spider, and it is that it's semi-social, meaning that multiple individuals build their webs next to each other. So they like to be next to each other, but they don't share a web. And this is quite cool. So you see, like in some populations, you can find like thousands and thousands. And these aggregations are really cool because you can find multiple color variations in the same aggregation. So here at the bottom right, you can see a black and white morph, here really tiny. And right at the back, there is an orange morph. So they basically share the same microhabitat. So first, I will show you what I found in the western part of Australia. So in this map, blue tones represent regions with high precipitation, and yellow colors represent dry areas. And when we look the color morph frequency to the morph frequency in space, the black and white morph is always the most abundant one. So you can see that this bit of gray is huge everywhere. And the black and white, the black morph frequency changes dramatically across space. And it seems to be more abundant here in this places with high precipitation. And as soon as you move to the dry areas of Western Australia, the orange morph starts to appear, increasing its frequency. And I observe a similar pattern in southeastern Australia, that is where I am right now. You can observe that the dark morph frequency is reduces. So here is really abundant, start being really common here. And as soon as you start getting into the desert in Australia, it is basically replaced by the orange morph, which was a really cool pattern to see on these maps. So to actually confirm our observations, we ran a multiple regression on distant matrices to test if these 6 variables could explain the change in morph frequencies across the space. So we calculated the morph frequency distances using Euclidean distances, and we also calculate the distances between localities under these environmental variables, also using Euclidean distances. And we found that precipitation was the only variable that partially explains the change in frequency of the color morph of the Christmas spider. So when we explore in detail using linear models the association of the frequency of each color morph with precipitation, there is no association between precipitation and the frequency of the black and white morph. As you can see, this line here is a little bit flat. However, when we explore the frequency of the black morph against precipitation, its frequency increases in places where it rains more, and it decreases in dry places. And the opposite pattern, even with a few, not as many points, uh, emerge from the data of the orange morph. So it seems to be more abundant in drier places and is almost absent in places with high precipitation. So this means that precipitation has a relevant effect on the frequency of the orange and black morph of the Christmas spider. This geographical pattern has been called Glogger's rule, which predicts that in places with higher humidity, animals should be darker. And it is possible that the presence of darker colors in humid areas provide resistance to pathogens or give camouflage protection. However, the mechanisms and the causality behind the Glogger's rule are still unclear for male lineages, and it remains to be explored in the Christmas spider. So from this part, we can conclude that our findings highlights the role of climatic factors in shaping phenotypic variation, including color polymorphism, and supports the ecogeographical association of dark coloration with humidity. On this part, there are a lot of other things to consider that we are still like exploring. So it seems that these different color morph varies on how they reflect the different light spectrum. So this is how they look to our eyes. They seems to be like really similar under UV. And when we consider the near infrared side of the spectrum, the orange individuals kind of like shine. So it seems that they're having some thermal benefits because of their coloration. But we need to explore this a little bit more. Also, we have a similar pattern when we took photos under different light spectra on the dorsal side. And I discovered that there are actually other morphs, so it seems that the color variation is not as simple as we thought from the beginning. But this color variance seems to be more abundant in the desert, so we need to go there and explore a little bit more. But what is behind the dark and white coloration on these spiders? But this, I mean, like, what is the actual difference in the structure and probably genes? So we don't know much about this. So we decided to start exploring, taking some microscopic photos. So we cut the spiders transversal at the bottom. So we did cuts here, and we put them under the microscope, and we found that the black and white individuals, have these like caves with some rocks inside that they are absent in the black individuals. And when we like zoom in on these caves, we found these beautiful shaped crystals of guanine that had been described before. So we know that the mechanism behind the white colors are guanine crystals, and probably the responsible for the dark colorations is melanin. This was really nice to see in the microscope. So taking advantage of the amount of eggs that we found in the field, we start seeing a lot of like baby spiders and eggs. Well, so we thought like, what if we try to grow these spiders in the lab, and during the development we can capture maybe some genes associated with guanine expression or melanin expression, and this can like guide us a little bit more to understand how this color variation happen and maybe what type of genes are involved. So I started growing the spiders in the lab, and it's really interesting to see how they grow. So they all look like this, like a little like brownish when they emerge from the eggs. After 7 days, they all have these dark colors at the bottom, and these white colors at the top. And after 40 days, some of them lose completely these white colors, and some of these continue with a similar pattern. And at this point, the phenotype is already on its way. And after a few months, they ended up being adults as we usually see them in the field. So we took samples of a RNA from these 3 stages. Unfortunately, I still don't have all the data. I sent it for sequencing like 2 weeks ago. So what I'm gonna show you right now is really preliminary. So a general principal component analysis of the gene expression pattern that we obtain seems that there are different genes involved in the different stages of development. So the PC1 splits this stage from this stage, and it seems that we capture maybe some genes associated with the color phenotypes. Probably they are here in these variants of the PC2. So vertically, we have this differentiation between color phenotypes on these different stages. So when we look generally at the pattern of expression of some of these transcripts, some of them seems to be overexpressed in the white phenotype, while they are like underexpressed on the black morph. However, I'm still annotating this transcriptome. There is no reference genome. So yeah, we know that there are some transcripts involved, but we don't know what they are yet. And when we took some SNPs out of these transcripts, it seems that we found what I just showed you is different in gene expression, but it actually seems to be some SNPs involved in this differentiation. So yeah, when we took SNPs out of the transcripts that are overexpressed, it seems that they could explain some of this difference in phenotype. So I plan to tell you more about this in Montreal if you guys are going there. So I'm more than happy to talk about this there, and hopefully I have a better story to tell you. I'm about to finish my PhD during this year, so if you want to continue exploring this, maybe collaborate, or if you hear about any job, please let me know. Before I finish, I want to thank my supervisors, Ileana Medina and David Stuart Fox, for all the feedback and support, and also to the Society of Study of Evolution that through the Lewontin Early Award made this possible, and is the reason why I'm here. Also to the University of Melbourne donors, the Arachnological American Society for the money to do the field work back home, and the Ecological Society of Australia to facilitate the exploration of the Australian habitats of this spider. And thank you all if you are still here, and I'm happy to take some questions.
All right, thanks a lot, Fabian. Great talk. Tons of work, a lot of driving. And
I love driving.
I forgot to mention in the intro to say thank you so much for staying up so late. So if anybody has any questions, I think it's approaching 3 a.m. for Fabian, but welcome to use this time for questions. Yes, Jessica.
Hi, Fabian.
Hey, Jessica.
Yeah, I have a question about your transcriptomics and if you use the entire body for your presumably RNA-seq libraries, or did you do it specifically from, say, the parts of the body where the color genes are going to be expressed?
So yeah, we wanted to use like the parts of the body, but we didn't get enough RNA, so we ended up using the whole spider. They are not that big. So yeah, maybe we catch something that is actually maybe not related with color there.
Yes, Masi.
Hi, Fabian. Great talk. I wanted to ask if you look at the different developmental stage of the black and white spiders using the microscope to see the photo that you show us.
So yeah, I was looking at them, but when they're growing up, we don't differentiate how they're gonna be. So that's why we decided to sequence when we actually could see the phenotype. So as soon as in that stage, that is after like a month, when we could actually differentiate, we decided to sequence them. But before that, we didn't know. We know that, for example, in a single exile, we can have both color morphs, but we unfortunately don't know anything about the inheritance and yeah, what is related with this color polymorphism.
All right, we have a question from a number here, but I don't know how to call on the name. So 03.
It's Bill Eberhard. I'm sorry about the number. Somehow the Zoom program has decided I only merit a number. I had a couple of comments. One, that in the first part of your talk, you looked at or noticed different groups that have color polymorphisms. And I think there is a common pattern that you didn't point out, and that is that in all of those groups, the spider sits out exposed on its web during the day. And many other orb weavers sit at the edge of the web, and that difference may be part of why some are have color polymorphisms and others don't. The second
would be nice. Yeah,
thing
I didn't discriminate between the biology of the spider, but that actually can make a lot of sense.
right, and there is a nice study in Brazil of I think a couple of species of Eustala, a genus that wasn't included in your list, in which they sit against trunks of trees and instead of out on the web, and they also have strong color polymorphisms that are associated with the kinds of bark and trunks that they're resting on. I'll send you a reference to this paper. I can't remember the complete reference at the moment. A second
Yeah, it
thing
would be nice.
that's interesting for you to think about is that a number of orb weavers that sit on their hubs at the hub during the day show clear signs of trying to avoid getting too hot. And the avoidance has to do with orienting the abdomen so that a minimum of the surface of the abdomen is exposed to direct sunlight. And that reaction has been seen in Nephila, it's been seen in Leucauge, and it's also been, I've seen it, I don't know if it's published, in Gastrocantha. In Gastrocantha,
There's a paper from the 60s that they reported that they ended up being kind of like diagonal
right,
to the
they
sun.
incline their abdomen, that's right. And so I think that there's a pretty clear evidence, at least under some conditions, that spiders out in sunny places try to avoid being overheated, and that would select against darker colors. And the final point, you were talking about your possible future. There is a very clear difference in Costa Rica between lowland Gastrocantha canthriformis and highland Gastrocantha canthriformis. And the tendency is exactly the opposite of the one that you found with your Christmas spider. That in the lowlands, white morphs are often common, and this is in strongly seasonal lowland habitats that have a strong dry season and hotter temperatures than in around San Jose, where it's 1,000 meters, where the white morph, which is the most common morph down near the Pacific coast, is now almost completely absent, and almost all of them are yellow. So, and that's cooler, and it's more humid. So you could
pretty interesting. I didn't know that.
you could think about applying for a fellowship at the Smithsonian Tropical Research Institute in Panama to work on this particular pattern that I know exists in Costa Rica, and I don't know in Panama what pattern there may be, but in any case, it's an interesting case because it runs exactly in opposite direction from the Australian spiders.
Yeah, thank you. No, that's really nice. Especially I really like Astracantha. Didn't know.
So
This
I'll send
changing.
you an email and spell some of this out.
Yeah, thank you, William.
All right, I just have a quick question. The black and white one, you showed two images, and one was definitely black and white, and one was a little yellowish. Is that a sex difference, or is that difference within the morph?
Yeah, it's something that I didn't highlight. It's like this color polymorphism of the Christmas spider is only on females.
Oh, okay.
The males, they are not reported as color polymorphic, but I found some variation that I didn't show you here. But there's also like it needs to be explored what is happening and going on with the males. But I will show you is like maybe the yellow thing that you're referring to is the ventral side, the ventral variation.
Oh, I see. One was the top and one was the, okay, got you.
Yeah, exactly. Yeah.
Okay, cool. And Dr. West Eberhard, you have a question?
If you have time for one more.
Yep, absolutely. Fabian's okay.
Fabian, you have all this richness of information on coloration in spiders, and it's such a mysterious polymorphism. My question is, so I'm looking at your graphs and the variables, the climatic variables you were looking at, all of them like temperature, rainfall or precipitation, radiation, all of those are continuous variables. And yet you have alternative phenotypes. You have a polymorphism, which means you have discrete morphs. And I was wondering why you would not observe continuous variation with temperature, like along lines, or if you have any ideas about why you have discrete color morphs.
I have no idea. It's probably if one of my guesses is that they need to camouflage really well. So with the background, but this camouflage hypothesis haven't been explored. And actually, I thought that I have like a continuous variation in the white proportion of the black and white individuals. But when I look up the photos and extract the proportion of white, it didn't vary. It's like continuously. So I reckon that maybe what is going on is that there is some benefit of having a certain morph, but besides the general geographical pattern, we don't know what else is going on if like a species of birds are discriminating between them, or yeah, there are a lot of possibilities. It would be nice to explore actually what they are not continuous at all.
Yeah, that makes sense what you're saying. I noticed that during, you had some nice information about development, and you had intermediates in the immature spiders. So that means that if it were advantageous to have a continuous variation, you could probably get it.
Yeah.
But Yeah, if you ever have any more thoughts about the discreteness, I'd be interested in those.
Thank you. Yeah, it would be nice to think about it. I haven't actually considered that question at all.
Thank you.
Yeah, thanks.
All right, everybody. I'm just looking in the chat for more questions, but we're right up against the hour here. I think we have like a minute left, and so I guess I'll thank Fabian for giving the seminar, and great talk and great answers to the questions, and we'll let you get some much deserved rest there.
No, thank you. I even forgot that it was 3 a.m. already.
I figured you could rest because of the time of day and giving the seminar, and just from all the driving from around Australia generally.
No, thank you all for being here, and yeah, thank you to the GSA and to you, Robert, for all the effort putting this together.
Thanks so much, everyone. See you next week. See you in Montreal.
See you.